This is the third lecture on enzymes, so I'll call this Enzymes 3. Now, for those of you who don't like math, there are some graphs here, but don't worry, they involve no calculations. You do not need a calculator. You don't need any math skills other than being able to understand what's going on here. So just kind of qualitatively. So what I like to talk about here are other factors that affect enzyme activity. If you remember from the previous lecture, and actually we'll talk about this again, one of the things that affects enzyme activity is simply the number of enzymes. So the enzyme concentration. Right, so the enzyme concentration, how many copies of the enzyme do you have? And we also talked about allosteric effectors. They can either increase or decrease the rate of a reaction. So we talked about allosteric effectors. We also talked about enzyme concentration, and we're going to look at this in a little bit more detail. That'll come up in just a moment. But first on this slide, we're talking about temperature and pH. So first off, most enzymes have a pH optimum that is around physiological pH. So when people talk about physiological pH, they're essentially talking about blood pH, which is actually a range. So I'm going to say it's 7.3 to 7.4. So sometimes you'll hear them report the average is 7.35. That's physiological pH. So that's what they're talking about here. Now, it turns out each enzyme has its own pH optimum. They're just showing that this enzyme works best, right, when the pH has to be 7.3. Now, the reaction rate, there's various ways of measuring reaction rate, but what I'm going to talk about is concentration of, generally you can either follow the disappearance of reactants or the accumulation of products. So since this is going up and products numbers increase their concentration. Okay, so the reaction rate we can measure in terms of the concentration of products. So concentration of products. So let me just, over here in this space, let me just very briefly talk about reaction rate. So a rate is a change over time, right? So how fast something is going, maybe like how fast you're driving down the road or running in a race. It's miles per hour. So it's some unit time. And for enzymes, we usually talk about it in terms of minutes or seconds. So here I'm going to talk about minutes. And what you'll notice is the product concentration. So we put products here in square brackets. So you might remember concentration is represented by square brackets. So this would be the concentration, the molar concentration, the moles of product per liter that are formed per unit time. So naturally, at the start of the reaction, there aren't any, and so it starts off at 0 and then kind of climbs like this. This would be the products. Right? Well, the reactants get used up. So I don't know, well, maybe we're here at, say, 1 molar concentration. The reactants get used up, and so they go down over time. And I'm not going to explain why they level off here. That's getting a little bit too much complicated for our needs, but they do level off over time. But products accumulate over time, and here we have reactants. Their concentrations decrease over time, and that's essentially what we're measuring here, is how fast they are going. So, all right, so what's going on here? Why they have a pH where they work best, but you get away from that too much, and what's the maybe this is about like 90%. Right, so there's a range here where the enzyme seems to work pretty well, maybe about 1 pH unit on either side, from like 6.3 to 8.3. But you get much beyond that, and it starts dropping off pretty fast. So, what's actually going on here? Well, you might remember how you get this is a low pH, so this is acidic. What happens when the pH gets too low? Well, remember one way to denature proteins. Enzymes are proteins. One way to denature enzymes is by decreasing the pH. So this can lead to denaturation, and that's the unfolding and precipitation of the protein, of the enzyme. and that kills it. You can also raise the pH. So high pH, this is basic, and you can also denature the enzyme by having a high pH. They actually mention this here with temperature, but the same thing can happen with either acidic or basic conditions. And really what's happening here is, if you remember, the acidic and basic conditions beyond pH optimum, this destroys those salt bridges, those ionic bonds that we talked about in the protein chapter between acidic and basic side chains. And that can happen with either acidic or basic conditions. All right, so you want to keep the pH near its optimum so it works good. And you don't want, and by the way, this denaturation, if you say you're starting off with the pH optimum and you start going to lower and lower pHs, once you precipitate here, once it's denatured, this is irreversible. Remember, so we can go down this way and get the enzyme working less and less efficiently. Eventually, you'll kill the enzyme and it can't come back. So if you want to raise the pH from this way, you're not going to be able to renature the enzyme and get it to work. Same thing on the basic end. So this denaturation is irreversible. Same sort of thing happens with temperature. So here they have the temperature. I'm guessing this is about 37 degrees Celsius. That's normal body temperature. And we're warm-blooded animals. Our enzymes are designed to work at this temperature optimum. If the temperature gets too high, right? That's what happens in a fever. I mentioned this. You start unraveling, breaking those hydrogen bonds. Break H bonds for hydrogen bonds. The protein denatures, and this is also irreversible. So it goes one way, does not go back. Once you denature it, it precipitates, and that enzyme, that protein, is done. This is a little bit different. You notice it's kind of, the curve looks a little bit different. That's not all that important, but the main thing here is that this is reversible. So one thing about any chemical reaction, whether it's enzyme catalyzed or not, is that it works slower at lower temperatures or faster at higher temperatures. The rule of thumb is the rate doubles for every 10-degree rise in 10-degree rise Celsius in temperature. So from 20 to 30, we should see, I don't know if this is 20, this is 40, we should see about a doubling for every 10-degree rise. That's just a general rule of thumb. But the main thing is any reaction, any chemical reaction, is faster at higher temperatures. Why is that? Well, two reasons. One, again, if the atoms or molecules have to collide, if they're moving around faster, that's what that means. When the temperature is higher, the molecules are moving around faster, then there's more collisions. So there's more collisions per unit time at a higher temperature between those molecules, and so they're more likely to interact with one another. Also, the energy of the collisions increase. So not only are there more collisions, the energy of those collisions increase. Collisions increase. And you remember something about activation energy? Remember that? So if the energy of the collisions increase, that means that more of these collisions have sufficient energy to get to this. More of these collisions have sufficient energy to get up to the activation energy, form that transition state, and continue on to products. So it's not surprising that we see an increase. But why does it decrease here? Well, because you start breaking hydrogen bonds and the protein unravels, and then eventually it dies and there's no more enzyme activity. One of the things I mentioned here is that this is reversible. So you can cool this down and warm it back up, and cool it down and warm it back up, and it's reversible. One thing you have to be careful about, though, is if you get to 0 degrees, all right, and we are mostly bags of water, right? So all these reactions are occurring in solution, so this is 0 degrees Celsius. Once that solution freezes, the ice crystals that form denature protein or the enzyme So you can get it cold, but if it freezes, you're going to lose some enzyme activity. So there's certain things that chemists will do to kind of keep it cold. Maybe even keep it around 0 degrees, but don't let these ice crystals form. And again, that's beyond the scope here. But that's one thing that people, you see in sci-fi movies and whatnot, where they talk about cryogenics and freezing someone, slowing all their reactions down, their metabolism down, kind of putting them in a hibernative state, and then years, maybe decades or centuries later, thaw them out and they come back to life. For some organisms, simple organisms, that's actually possible, but for more complicated organisms, it's not, and that's because these ice crystals form and denature the protein, and it basically turns to mush. So we talked about temperature and pH. Next we'll talk about, I think we're going to talk about enzyme concentration next, and then substrate concentration. Oh, not quite yet. Something more about physiological pH. So not all enzymes work best at physiological pH 7.35, and the reason for that is, although our blood is, does it say here? Where's blood? Blood plasma interstitial fluid 7.4, 7.35. That's not the only fluid in our bodies, and these different regions in our body, these different solutions have different pHs. So pepsin, for example, is located in the stomach. It's a digestive enzyme. It breaks these peptide bonds. It's responsible for digesting proteins. Its optimum pH is pH of 2. And, well, why is that? Because it's in the stomach, where the stomach pH is 2. So if it's going to be digesting proteins in the stomach, it better work best at pH 2. Kind of a contrast to that would be trypsin. This is also a enzyme responsible for digesting proteins, right? Peptide bonds. Talked about trypsinogen gets converted to trypsin, pepsinogen gets converted to pepsin. These zymogens get converted to their active form, so they're digestive enzymes produced as zymogens, and then here's the active form. Well, this is in the small intestine, and the small intestine actually has a basic pH. Right? Well, why is it a basic pH? Well, if you know your anatomy, the stomach, pH of 2, empties into the small intestine, and so the small intestine has a higher pH to kind of buffer and neutralize that acid that comes from the stomach. So this works best in basic conditions. Likewise, arginase is in the liver, very high pH optimum, 9.7. Some of these others not quite as noticeable, and again, that's because all these body fluids have different, and actually talks about this. It says body fluid, but a couple of these, the lysosome, for example, this is an intracellular organelle. So if you remember your cell biology, we have a nucleus, for example, where the DNA is kept. The lysosome is kind of like the stomach for the cell. So lysosomes have a low pH, so the enzymes you find in the lysosome are going to be lower, right? Gastric juice, and again in our stomach, so these digestive enzymes in our stomach are going to work best at low pH. Pancreatic juice, again, the pancreas dumps into the small intestine. So the main thing here is to recognize that not all body fluids are at 7.35. Urine, notice there's a huge range here from 5 to 8. Now that's only 3 pH units. So I'm going to write here 3 pH units. But remember, the pH scale is logarithmic. So from pH 5 to 8, this is 1,000-fold difference in acidity. Right? It's not just a difference of 3, it's a difference of 10 to the third, so 1,000-fold. So there's a large range here. And the reason for that is urine is partly responsible for balancing our physiological pH. So if our blood is too acidic, it'll dump some of that extra acid in our urine and get rid of it. If it's too basic, it'll keep some of that acid and lower the pH. So I'll just say the kidneys, where the urine is made, kidneys help regulate blood pH. And so that's why you see a large range here. Urine isn't necessarily acidic or basic. It depends on the kind of food you're eating. It depends upon how much exercise you're getting, and that sort of thing. And the last thing I'll mention here on this is this: beware of scam pH acids bases. This is the same guy, I believe, that pH balanced diet fella. Oops. But we talked about him right at the very beginning of the class. They promote an alkaline diet, and it's really snake oil. It's not real at all. So here's a website where they're trying to tell you you must have alkaline water or alkaline food. It's hooey. Your body will take care of itself. The diet is not going to affect physiological pH. Well, actually, it will if there's something wrong, but it's not going to. You're not going to fix something by having an alkaline diet. All right, so here's the effect of enzyme concentration on reaction rate. Again, the reaction rate we're talking about. Okay, so let me draw this. We're talking about reaction rate here. Let me talk about, again, how we measure reaction rates. So we have the following accumulation of products over time. So this is a concentration, say a molarity of products, and it accumulates over time, and then it kind of levels off. And so that was product concentration. And the reason why it levels off is because, remember, the reactants are getting used up. So as the reactants concentration goes down, the product concentration kind of levels off because we don't have enough reactants to make any more. So the concentration stays constant. So the rate that we're talking about is any point along this line. It would be the concentration per unit time. That's what we're looking at here. So concentration per unit time. So this is kind of an easy graph to recognize. Essentially, what we're looking at here is a linear response. So if we, let's call this, we'll just put in, these would not be molar concentrations. Typical concentration might be micromolar. This is 10 to the negative 6th molar. So 1, 2, 3, 4, 5, 6. And what I wanted to point out here is if we double the concentration, we double the rate. All right, we started off at 1, we went to twice as much enzyme, now we have twice the rate. 2, 3, 4. Right. If we double the concentration again from 2 to 4, maybe this isn't quite right. It would be helpful if I had graph paper. All right. So a doubling, essentially a doubling of the enzyme concentration doubles the rate. So it's a one-to-one correspondence. Think about what's happening here. You have an enzyme binding a substrate, going to the enzyme substrate product, and then going back to free enzyme and releasing that product. Well, if we had twice as much enzyme, so not just one but two of them now, we would have twice as much enzyme substrates products and then twice as much product. So every time you double the amount of enzyme, you double the amount of product. So there's two ways in which we can increase enzyme concentration, which we've already talked about. One is de novo synthesis. This involves transcription of the DNA to RNA, DNA to messenger RNA, and then translation. I won't write the whole word out, but translation to make the final protein, and then maybe there's some post-translational modifications that we talked about with prosthetic groups or glycosylation or proteolytic cleavage or whatever, but this takes a while. It's slow. Zymogen activation, remember there's two things we talked about it. First, it's fast. Well, three things. One, it's fast, so you can make a lot of this protein quickly. Two, it stores these potentially deleterious enzymes in a harmless form, so in a way that it won't hurt the cell or the organism. And three, looking at the clotting cascade, it allows us to amplify a signal such as you see in the clotting cascade. So with zymogen activation, we get a very rapid increase in the enzyme concentration, so we can make a lot more product per unit time. The effect of substrate concentration. So we talked about, let's review here real quick. We talked about pH. We talked about temperature. Enzyme concentration, And now we're talking about the substrate concentration. So the substrate concentration, right? So here you see a leveling off, and this is the reaction rate, and we could be following accumulation of reactant or accumulation of products, or we could be following the disappearance of reactants. It doesn't really much matter, but we're looking at the reaction rate here again. So this is the change in the concentration, say a molar concentration per unit time, say per minute. That's the rate. And it increases almost linearly as the substrate concentration increases, at least for a while, and then it levels off. We get to a point where it levels off, and they call this maximum activity. This is also known as Vmax. Vmax here. It stands for velocity, the maximal velocity. I want to take a break here for just a minute, and before we talk about Vmax, I want you to see this quick video clip.
तोड़ना उसका उंगला बसा के
हाँ जी
अल्लाह दिखाओ कि मैं फूट गया $100 सुट्टा मैंने सेंट है सारा पैड.
So what did that video clip have to do with enzyme activity? Well, what I want you to think about here is, think of the enzyme right here. Think of the enzyme as a fruit picker, or that. Okay, when they're working as fast as they can, what that means is all the enzyme is combined with substrate. The substrate concentration is so high, right? Say 40 micromolar or something like that. The enzyme concentration is so high that all the enzyme is essentially bound up with the substrate. It's similar to a fruit picker where the grape vine or the apple tree or whatever it is so full of fruit that their hands are essentially, if you want to think of them as the active side, as they reach in and grab a hunk of fruit and put it in the basket, essentially their hands are constantly carrying fruit. As soon as they drop it in the basket, they reach into the plant and pick up another hunk of fruit, and their hands are constantly occupied with fruit. That's what we see up here. Down here, so this up here is where we have a lot of fruit on the tree or on the vine. Down here we don't have a lot, and so the fruit picker is kind of hunting around and looking and trying to find the next apple or the next hunk of grapes to pull off the vine. So there is a significant portion of time where the enzyme is empty. It's not occupied with substrate. So we say here the enzyme is combined with substrate. We say the enzyme is saturated. Enzyme is saturated with substrate. And it wouldn't matter if there was more fruit in the tree. They're not going to be able to pick any faster because their hands are already occupied. Down here, the enzyme activity, well, it says enzyme activity increases. You can see that for yourself. But the enzyme is, I'll just say, is often empty. What I mean by that is not bound or not binding substrate. So this is a case where the fruit picker is hunting around and spending its time looking for some substrate and not actually catalyzing the reaction. All right, so Vmax is a maximal velocity. This is as fast as the enzyme can go, and at Vmax all of the enzyme is bound up with substrate. Okay, we say it's saturated. It can't go any faster. The active site is essentially always has a substrate in there. Then there's this other value, Km, and I had mentioned this before when we were talking about inhibitors. There's a V effect or a V type and a K type of allosteric regulation, and this K stands for the Michaelis-Menten constant. Michaelis-Menten constant. I mentioned there that it is essentially a measure of how well the enzyme binds to substrate. All right, so how tightly the enzyme binds to substrate. Let me erase this a little bit here. So in talking about Km, another way to think about Km, actually for this class, this is the first way here is fine. Another way, which I'll explain, is this is the substrate. The Km is actually uses units of concentration, so molarity, but it's the it's the substrate concentration. And we use square brackets to denote concentration, so we read this as the substrate concentration that gives 1/2 of the Vmax. So what the heck are they talking about here? Well, here's the Vmax. Say this is about 1/2 Vmax. I don't know what this value is, but this is about half of it. Okay. The substrate concentration that gives 1/2 the Vmax here. Let's just give this a value. This is about 10, so let's say this is 5 micromolar. Let's look at a different enzyme. So there's an enzyme in blue here. Let's look at a different enzyme. Let's say its substrate reaction curve looks something like this. It has the same Vmax. It doesn't actually have to have the same Vmax, or whatever. It has the same Vmax. But notice, to get to 1/2 of these Vmax, we have, I don't know, this is 2.5, this is, I don't know, maybe 0.5. It's like 10 times lower concentration than this one. 0.5, 0.5 to 5. So this one in red here binds the substrate a lot better because we get to 1/2 the Vmax with only 1/10 as much substrate. So that's why we refer to this Km as a measure of how well the substrate binds, and it's usually expressed in terms of substrate concentration. So 0.5 micromolar versus 5 micromolar. Okay. We could look at another enzyme. Again, we use the same Vmax, but maybe its curve kind of runs off like this before it levels out. So to get to 1/2 the Vmax, we have to get to a much higher concentration, so 20 micromolar. Right? This blue one, this enzyme binds weaker or with less affinity, and it has a higher Km. So another thing to mention here about Kms, all right, the lower the Km, the lower the Michaelis-Menten constant, the Km, the tighter the enzyme binds the substrate. So getting back to those K-type and V-type inhibitors. K-type inhibition, they both slow down the enzyme, but for different reasons. K-type inhibition affects how well the substrate binds, and that's the example that they used in the book where they showed the allosteric inhibitor binding someplace, right? Not binding the active site, but binding somewhere else, but closing down that active site, so it made it harder for the substrate to bind. All right, well, that would mean you need a higher concentration of the substrate to kind of force it in there, so you'd have K-type inhibition, right? It would have, we could use the same sort of thing here with, if this blue line here, this is the normal enzyme. If we added a K-type inhibitor, okay, instead of being at 1/2 Vmax at 5 micromolar, maybe we had to increase that concentration fourfold to 20 micromolar to kind of force that substrate into the active site. Okay, so that would be K-type. So maybe we can think of this as K-type allosteric inhibition. Now with the K-type allosteric inhibition, what that means is you can overcome that inhibition by adding higher and higher concentrations, right? You can eventually get to the Vmax, you just need a higher concentration of substrate. With a V-type of allosteric inhibition, that was the case where the substrate could still bind, but maybe the inhibitor affected the ability of the enzyme to undergo a conformational change to close down on the active site and actually catalyze the reaction. So the substrate still binds fine, but the maximal velocity is reduced because we've affected the actual catalytic step. So let me get rid of this line, and a K-type inhibitor would have the same curve here. We'd still get it would not affect the Km. Okay, so we would still get to the Km would still be 5 micromolar, but its maximal velocity would be less. So what would that look like? Okay, I don't know if I did this quite right, but here's a Vmax. So this is that same blue enzyme here, the same enzyme we have here, but this is a Vmax. This is now plus inhibitor, and it's lowered the Vmax. So now the Vmax is here with this K-type inhibitor. So what's the 1/2 Vmax? I kind of missed it a little bit. Let's be down here more, but let's say it's about here. Can we redraw that line a little bit, smear it out? Okay, so 1/2 Vmax, we still have the same Km. It's just the velocity has been decreased. Okay, so that's a V-type allosteric inhibition. The thing about V-type is it doesn't matter if you add more enzyme. You're not ever going to be able to overcome this inhibition because you're never going to get up to the maximal velocity. So this V-type inhibition cannot be overcome by increasing substrate concentration. However, with the K-type, yeah, it affects, that's this blue line here, it affects the enzyme, how well it binds, but if you add enough substrate, you can actually still get up to Vmax. So with this K-type, this can be overcome by adding more substrate, or I should say by increasing substrate concentration. Okay, isozyme. All right, we talked about isomers, all sorts of different isomers. We had structural isomers and stereoisomers and enantiomers, mirror image isomers, and whatnot. Iso means the same. So these are enzymes that the thing that's the same about them is that they catalyze the same reaction. So you have the same reactants, same products. So lactate dehydrogenase converts lactate to pyruvate, and there's 5 different enzymes that do it. So lactate, right, this is, lactate is, lactic acid would be protonated. Lactate is the ion. So this is lactate. It's an alpha hydroxy acid. And this enzyme, lactate dehydrogenase, converts it to pyruvate. So this is a reaction that's carried out. Now you don't have to know about the structure of lactate and pyruvate yet. You will in the next chapter. That's a big part of the next chapter, 2 key molecules. So here we're oxidizing the alcohol to the ketone. So this is alpha keto acid. This is pyruvate. And lactate dehydrogenase, this is an oxidation, right? So dehydrogenases use NAD, converts NAD to NADH. The alcohol is getting oxidized, NAD, the coenzyme is getting reduced to NADH, getting a hydrogen as a reduction. All right. Anyway, so the thing is there's 5 different enzymes that catalyze this. And what we mean by that is, okay, they're catalyzing the same reaction, converting lactate to pyruvate, but these different enzymes are coded in different genes. that means they have different DNA sequences, which means they're going to have, so it doesn't say this here, but different DNA and amino acid sequences. So that means that even though they have different DNA sequences, their primary, secondary, and tertiary structures are going to be very similar because they have to carry out the same reaction, but you can still distinguish one from the other. Because they are, we use the term expressed, they are produced in different tissues and different organs and different cells. We can use the type of isozyme that we find to kind of tell you what kind of organ and what kind of cell, what kind of tissue has been damaged or diseased. They're the same enzyme in that they catalyze the same reaction, but they have different DNA sequences, different amino acid sequences, different tissue expression. Just real briefly what we mean by expression. You might be familiar with like all your cells, except those that have lost the ability to replicate, have lost their nucleus. And there are some like that, like red blood cells, for example. All cells have the same DNA in them, okay? But clearly, the cells in your muscle, is different from the cells in your lung, is different from the cells in your liver and whatnot. So that's what we mean by different tissue expression. Some of these DNA sequences get translated into protein in one tissue but not the other. Now the cells, regardless of what cell they are, they still need to be able to carry out this reaction. They're just going to do it with different enzymes. Okay, so all they're showing here is, okay, you have isozyme and a muscle isozyme. Now the lactate dehydrogenase turns out is a tetramer, right? So well, let's just look at here. So in heart muscle you just have this H4 isozyme, so it's an H4 tetramer. All four of these are the H type. In skeletal muscle, you have the M type, all four of the M type. And there's 5 different varieties that you can have here. So depending upon, and again, these are all different tetramers, right? So here we have an even numbered one. Here we have only one. We can use this to kind of figure out what tissue was damaged. So if we find this type circulating in the blood, we know you have a problem with your heart or your kidneys. That's for lactate dehydrogenase. But there's others as well. So creatine phosphokinase, we find that in the heart, and so when heart tissue is damaged, indicates you had a heart attack. If you have liver or bone disease, you'll kind of find an alkaline phosphatase and so forth. So we can use these isozymes. Well, here they're talking about isozymes. Here they're just talking about other general enzymes that are used for diagnosis. These are not isozymes. But the isozymes that they have here for lactate dehydrogenase, again, there's 5 different types, 1, 2, 3, 4, 5 of them. These are not isozymes, but nevertheless, if we find these in the blood, we can use them to diagnose what kind of condition you had. So let's look at the last slide here. So following a heart attack, we're looking at creatine kinase. Okay, so this is following heart attack, myocardial infarction. CK is that creatine kinase from that last slide found in the heart. LDH we already talked about, lactate dehydrogenase. We talked about lactate dehydrogenase. AST is aspartate transaminase, which they didn't mention. And this is found in both the liver and the heart. So right after a heart attack, you see creatine kinase levels very high, and they drop off. This is in the blood, right? This is not where you want to find these enzymes. What this means is the cells died, they broke open, they spilled their guts, and their creatine kinase came out. So one thing you can do is trace how high these levels are, and you can tell the severity of the heart attack based upon blood work. So that'll do it for the chapter on enzymes. Next we'll be talking about metabolism and how the cells use these enzymes to convert food into energy.